We're honored to have Dr. Waitzman come back to Northwestern and be with us. I'm not nearly as good at doing these. Dr. Zakova said she wouldn't be here, and Dr. Quagan does a phenomenal job as well. But they roped me into it, so I assume they wanted to see my slides again. So when Josh last left us, it was in 2019. So what was going on at that time, right? It was just before COVID, and I think that might have been some higher power's way of saying maybe you shouldn't have left Northwestern. But we'll pull that aside for a second. So Dr. Chima's always had problems, with assessments. It's hard to do assessments. And so the Quagan lab developed a lot of different things. I have those on other slides that I won't show you, like knockout mice. And anyway, one of the things that came out was a hammer that the fellows had to lift up to prove they're worthy. And it was called Nephnir. Are you still using this as an assessment, Dr. Chima? I don't know. Anyway, so the fellows would get together and they would hang out, and then Dr. Chima would fly in and drop the hammer down, and the fellows have to pick it up and prove their worth. And so anyway, there's Alex Leiter. All right, sorry. All right, so more seriously now, Josh went to Beth Israel and had to join their fellows. And these are their fellows in general, and I think you compare our picture to theirs, and I think ours look a little healthier and better. Maybe Zach post-call might fit real in here, but in general, it fit better. So Josh joined them, and he was worthy, and he's done great. All right, so he was at Northwestern, right, 2007?
Yeah.
As a medical student, MD, PhD through 2019. So for 12 years was here, longer than a lot of people in the room. And then, due to life circumstances, he had to go to Boston. And so he joined Beth Israel in 2020. But he still bled purple because he helped us recruit Luke Barr from afar. So, huge. And then he's on staff as of 2021 at Beth Israel Deaconess Medical Center, got his KO8 in 2022, but still has time, despite raising a 9-year-old, to do a lot of other stuff. So he's in this Freely Filtered podcast, has phenomenal titles. He works for NEFJC with Jenny Lynn. He was on the ASN Workforce and Training Committee, and he's still clinically active. So he does a lot of cool stuff with his time. But the interesting thing, his research is on the structural, I just quoted this from his thing, structural biology and biochemistry techniques to determine how high-risk APOL1 proteins cause progressive kidney disease. So hugely important for us, right? Because we have Natera tests, we have Invitae tests, and this is one that I got in the last month. It's pretty obvious, but Lonnie had to help me interpret this. It's a positive test, but I was like reading down here and over interpreting it, I think. But we're getting these tests all the time. And so hopefully, Josh, your research is going to take us into a point where we get to some therapeutics to address this. So needless to say, we're all excited to have you back. Without further ado, we'll let you come up here. I just have to figure out how to get your slides back on. Do you know how to do it, or? Thanks, Marissa.
No, thanks, Marissa. All right, I don't know that this is doing anything. Can anyone hear me okay? Okay, excellent. So thank you, Jay, for that kind introduction. I hope no one tweets the picture of Beth Israel follows. I'll get there in a sec, sorry. Okay, so obligatory COI slide here. And before I talk about real research stuff, I just want to say how happy I am to be back. Jay outlined the length of time I was here. That's me on the last day of medical school. You can tell by the giant smile. And then was super lucky to get to stay here for residency and fellowship. The guy on my shoulders is now a 9-year-old. I just picked some of my favorite pictures from being a fellow here. Including the one of me and my classmates holding Jay up. Some great presentations here at ASN of a blank poster, and some some freely filled, Nef Madness draft picking with the Flaming Hot Cheetos. Okay. But today I'm going to talk more about research stuff, and I want to talk about four related things building on each other. I'm going to review some of the ways that we've identified this gene called APOL1 and how it has been associated with different kinds of kidney disease in people of recent African ancestry. And then identify some open questions that we still have, even though we've identified this gene over 10 years ago. Bullet number 3 is where I talk about the research stuff, and that should sound like gobbledygook at this point, but the goal is that you will leave with an understanding of a little bit of those two techniques, really trying to figure out how APOL1 works, and driving at an idea of APOL1 working like an ion transporter ion channel, and then identify some next steps of where I think the work's going from here. So this is a group that I don't need to tell about the disparities we see in healthcare and the disparities we see in kidney disease. Whether we're in the clinic, the dialysis unit, in the hospital, we know we see more people of recent African ancestry with advanced kidney disease and end-stage kidney disease than any other ethnic group or other group that we take care of. This is borne out by the data, and this is old data from the USRDS, but the trends still hold true. 2 notable things. Number 1, we're doing better at taking care of kidney disease. Those rates are going down. That's a good thing. But there are still significant disparities in the rates of kidney disease in those who identify as Black and African American, to any other ethnic group that we take care of. I think there are a lot of potential reasons for this. And we know that structural racism and disparities in care and social determinants of health are hugely impactful on outcomes and drive many of the worst outcomes we see in Black and African American communities. At the same time, I would argue that those disparities alone don't explain the scope of this kidney disease problem. I wanted to highlight a couple of comparisons between those who identify as Black or African American and folks who are Latino, another underserved group of color in the United States that has also been marginalized. And we can see that despite similar rates of diabetes, similar rates of access to care, here using health insurance as a proxy for access to care. Oh, sorry. There's no, sorry, let me take a step back. The rates of diabetes in those who are Hispanic and those who are African American are pretty similar, and yet we see huge increases in the rates of kidney disease in those of recent African ancestry. Using health insurance as a proxy for access to care, if anything, those who identify as Black or African American have better access to health insurance than do folks who are Hispanic, but again, have higher rates of kidney disease. And finally, blood pressure. I think a lot of us have grown up with this idea of high blood pressure drives kidney disease, and we see this especially in our patients who identify as Black or African American. And you would think that if we did a really good job controlling blood pressure, we would make a really big impact on this type of kidney disease. And unfortunately, that hasn't been borne out by the evidence. The African American study of kidney diseases and hypertension randomized people who self-identified as Black to a standard blood pressure target or a lower blood pressure target. And even doing a really good job of treating blood pressure here didn't make an impact on the rate of kidney disease progression. So I would say while these social determinants of health are hugely important, they don't tell us the whole story of the disparity we see in kidney disease. With this background, folks began to look for other explanations as to why there was this significant difference in rates of kidney disease. And about 15 years ago now, in 2008, a genome-wide association study identified a highly significant linkage between this place on chromosome 22 and the phenotype of non-diabetes associated kidney disease in Black and African Americans. Further fine mapping of that genome region done by Martin Pollack and David Friedman and colleagues narrowed that region even further to changes in the gene encoding a protein called apolipoprotein L1. And before we dig into this, I think it's helpful to figure out what apolipoprotein L1 actually does. This is a picture of a trypanosome brucei that causes African sleeping sickness. And the trypanosome lives by endocytosing stuff from your blood. The APOL1 protein hangs out as part of the HDL complex. And when the trypanosome endocytoses the HDL complex and moves that particle into a lysosome, the apolipoprotein L1 does something, causes lysosomal swelling and parasite death. And this is an innate immune defense way we have to protect ourselves against African sleeping sickness. Over time, we see the evolutionary battle between people and parasites go back and forth. And so at some point in the last 20,000 years or so, trypanosomes were able to develop a new way to resist this mechanism of trypanosome killing, and so became essentially immune to the original variant of APOL1 called the G0, or in this picture it's called wild type variant, and got much better at getting people sick. Populations that were exposed to high rates of trypanosomiasis developed variants in that gene of their own. We call those the G1 and G2 variants. I'll show a picture on the next slide. But those are really supercharged APOL1 proteins that are better at killing the trypanosomes that are now in circulation. So whereas the G0 wild type variant doesn't kill trypanosomes very effectively, these new variants, the G1 and the G2, cause lysosomal swelling, and then these parasites go on to die later. And because the prevalence of trypanosomiasis is so high in West Africa, that's where the selective pressure to develop these gene variants came from, and where these gene variants became common. With lots of very complicated history in the slave trade, folks who trace their ancestry back to West Africa have much higher rates of carrying these gene variants, and it's thought that that drives a large part of this kidney disease risk. And that's this slide. So here you can see actually the different variants in the apolipoprotein L1 sequence. These changes are pretty small on an amino acid level. The G1 variant has 2 single amino acid substitutions. The G2 variant has a 2 amino acid deletion at the end. These don't feel from a protein perspective like they should be hugely impactful. But then again, we'll talk about this, we don't totally know what this protein does. And the presence of one of these 2 variants, the G1 or G2 variants, provides a heterozygous advantage and enhanced ability to kill trypanosomes. But that enhanced ability, what did I do? Okay. But that enhanced ability to kill trypanosomes comes at a cost. And that's if folks have 2 copies of a high-risk variant, 2 G1s, 2 G2s, or a combination of a G1 and G2, they have a substantially increased risk of non-diabetes associated kidney disease. That's not great. Otherwise, you're going to stare at a black screen for a long time. Okay, great. So over time, more genome-wide association studies have been done on different types of kidney disease and have found strong relationships between these gene variants and different types of non-diabetes associated kidney disease. I think we tend to think of APOL1 associating with focal segmental glomerulosclerosis, a pattern of kidney injury we see on biopsy, but that's not the whole story here. We see substantial risk multiplier effects on APOL1 driving HIV-associated kidney disease, on driving lupus-associated kidney disease, and on driving the end-stage kidney disease we used to call due to hypertension, but now we should probably call hypertension-associated kidney disease because a lot of it is driven by APOL1 genes. I think we're also seeing these gene variants enter clinical practice. Like Jade had talked about, these are on the Natera panel. We get these results in clinic all the time. And although we don't have genetic targeted therapies yet, we're close to seeing them come out, and we'll talk about this a little bit more. So we've known about these gene variants for approaching over 10 years now. How do these gene changes drive disease? And I like this figure because it's kind of so terrible. Anytime you see like 20 arrows pointing to stuff, you know that we don't really know what's going on. So we don't really know what's going on. I'll note a couple of things here, though. Lots of different parts of the cell are thought to be involved in driving, have been invoked as driving part of this kidney disease, the plasma membrane, the mitochondria, the ER, to name a few. There are also lots of different cell death and degradation pathways that are invoked here. You can see autophagy, pyroptosis, and proteasomal degradation. And this is really a podocyte-centric view of the universe, which I think is a good view of the universe, but is not the only way people think about this kidney disease. Doesn't include endothelial cells, doesn't include circulating immune cells as well. And I think those are coming to the fore as playing a role here too. But over time, most people seem to agree that the APOL1 protein is able to form ion transport capable channels at lipid membranes. Whether those are the plasma membrane, the mitochondrial membrane, somewhere else, I'm kind of agnostic to. But this activity seems important for how the APOL1 works in trypanosomes and may be important to us in the progression of kidney disease. We've known about this even before we knew APOL1 had anything to do with kidney disease. When folks identified APOL1 as a trypanosome lytic factor, they actually associated it with membranes and looked at the ion currents that it was able to allow across those membranes. And you can see here that they actually thought it was a chloride channel to start with. Subsequent reports and really a large bulk of data supports that APOL1 can actually transport cations, sodium, potassium, calcium, in a non-selective way. And there are not a lot of ion channels that can transport all the ions all at the same time. I think we still have really open questions as to which of these ion conduction activities are real. We also know that APOL1 has a unique or interesting mechanism of activation. You can see here lipid bilayer experiments where they flow in APOL1 proteins at a low pH in the 5s and don't see any change in ion transport until they increase the pH to over 7. And that's when you see a non-selective cation current emerge. That's a reversible change with pH changes. And it's led to a model where we think APOL1 activates in at least 2 steps. A low pH membrane association event and a pH increase activation type event. And that is required for the ion transport that's way upstream of cytotoxicity. Although we have information about this multi-step process, we don't have the kind of information that ion channel people normally have. How quickly do these steps happen? How many molecular steps are involved in this process? And so I think questions about how quickly the APOL1 current activates and inactivates are still really open to us. And then what makes the high-risk variants high risk? How do those small amino acid changes drive disease? To date, there's only been one report on the effect of those protein changes on protein function. That's one group that looks at ion transport in liposomes and lipid balls. And they see that the G1 and G2 variants can transport about twice as much cation current or cations across the membrane than can the G0 variant. It would be really nice if someone could confirm this or refute this, but this is really the report that we have. And so I think it's important for us to understand, do these high-risk variants really allow for increased cation conductance? And if so, how do they do that? How do those small amino acid changes allow for a significant change in activity? Finally, the last piece of evidence that really cements this ion transporter role as critical to driving kidney disease comes from a phase 2 trial that was recently published in the New England Journal. The Vertex pharmaceutical company developed this small molecule called inaxaplin, and they showed that it's able to prevent ion transport mediated by APOL1. This is a picture showing you that when you express APOL1 in hex cells, you get a significant increase in thallium flux. Thallium looks like potassium to ion channels, so this is thought to be monovalent cation changes. And then when you add the inaxaplin molecule, you get a substantial reduction in that current, suggesting that it's blocking some feature of that ion transport. Why is that exciting? Because we see it make a difference on clinical outcomes. In a trial of that medicine in 13 patients, granted small number, they saw a 50% reduction in proteinuria over the course of 3 months. And this proteinuria reduction was at least somewhat preserved after the medicine was taken away. So this could be a disease-modifying therapy that targets this particular activity of the APOL1? There are lots of open questions here. How do we know that this is really an ion channel blocking molecule if we don't really know that this protein is an ion channel? And where on the molecule does it bind? Furthermore, questions about is it really specific for the G1 and G2 variants are open. And I think questions about are there other ways to block this activity of the protein? If someone has resistance to this one molecule, what are we going to do going forward? Although it blocks thallium flux, it doesn't mean it doesn't block any of those other ion transport activities either. And I think understanding which of these ion conduction activities is important to the progression of kidney disease is important to think about as we move new therapies to the clinic. Having structural information would make these questions a lot easier, but to date, no one has an APOL1 protein structure. And people have used different approaches to try to figure out what that structure might look like. One group that does chemical labeling thinks that it has 4 transmembrane passes, with one of them contributing to the pore. Another group that uses antibody labeling to specific parts of the protein thinks that it only has 2.5 membrane passes. And I think both of these can't be right at the same time. So there are clearly different structural models and have different implications for how the protein might work. This protein is also a small protein. It's around 40 kilodaltons or so. And most ion channel type proteins have to be bigger or have to oligomerize to be able to allow ions to flow through them. So although most people think these proteins oligomerize, no one's ever seen that for real. So understanding the topology of a single APOL1 subunit and how many of those subunits work together is another open question here. So recapping all those questions, does APOL1 allow for transitive cations, chloride, or both? How quickly does it open and close? How does the high-risk variant differ and drive kidney disease? And what's the structure of an APOL1 pore look like? And I think underlying all those questions is a clinical one. What APOL1 activity is it that we need to block to slow the progression of kidney disease? Questions at this point. I know I covered like 15 years of science in about 15 minutes. It would be reasonable to have questions. I am also comfortable with long, awkward silences. Dom, is that a question or is that a swatting a fly? Yeah, so animal models are challenging because this is a human-specific protein in trypanosome defense. There are a number of published animal models, including a mouse model developed by the group that I'm working in with Martin Pollack. That has shown a number of different things, but the disease that the mice get is really kind of a different one than the one people get. It's much more like fulminant all of a sudden, as opposed to slowly progressive over time. So as less of a mouse model person than some people in this room, I think it gives us some insight, but I don't know if it tells the whole story there either. Yeah. It was people with biopsy-proven FSGS, genetically confirmed 2 APOL1 high-risk alleles, and I think a gram of proteinuria or more to start. Mm-hmm. Right, and so we're waiting for the larger phase 3 trial, which is ongoing, to give us a sense of what kind of remission we see, or if we see remission in that group on that medicine. Yeah, I think one thing that's interesting about that trial is that it really took people at all stages of disease, from GFRs of 30 and proteinuria up to GFRs that are much better. And I think we may not expect to see all complete recovery in someone who's had that degree of baseline kidney injury, but slowing progression would be a huge win here. And I think I'm hopeful that the phase 3 trial gives us more information. There are also other companies that are developing small molecules in this space. But yeah, I don't know any of the answers to that at this point. All great questions, thank you. Okay, so with those open questions in mind, I wanted to turn to the work we've been doing to try to answer some of those open questions. And I've told you about a couple of different ways people have assayed these ion transport activities, and each of them has strengths and weaknesses. The liposome lipid ball method uses a chloride-sensitive electrode and is pretty slow and finicky to work with. The planar lipid bilayer stuff that's given us a lot of information about protein activity is very slow to work with. You can do like one protein a day. And also gives us steady state measurements. It doesn't answer those opening, closing molecular change processes that we're interested in. And patch clamp is probably the electrophysiology people. Most people at least have PTSD from medical school learning about. And this is a really powerful technique. It can be done on cells that express your protein. It's especially good to do on cells that don't die when they express your protein, but the APOL1 really quickly causes cell death when you overexpress it. So it's been a challenge to get good patch clamp data here. We at BI were able to do some patch clamp work that just got published earlier this year. I'm not going to torture everyone by introducing another technique, but even still, those were pretty small numbers of patch clamp cells, and we still have open questions from that work. And it's on to that stage that I want to introduce the fluorometric stopped flow method. This is a way that lets us assess how quickly a molecular process starts or stops. This is a schematic of the machine, and I think of it as a rocket-powered mixing device. You have a drive ram that's powered by like a compressed gas that mixes two things really quickly and then squishes them into an optical cell that you can read out fluorescence from. In the experiments I'll talk with you about today, we have a fluorescent molecule in the A syringe and a quencher molecule that turns off the fluorescence in the B syringe. And for schematic purposes, green shining A is fluorescence on, gray dark A is fluorescence off. And we can use this to study ion transport. We can pack fluorescent ants molecules inside of liposomes. We can load those liposomes up with transporters in their membranes. And if we activate them in the presence of thallium outside the liposome, we allow normal ion transport to happen. Potassium can go down its concentration gradient out of the liposome. Thallium can go down its concentration gradient into the liposome and quench the ant's fluorescence over time. This method is liposome-based, which I like because it's purified proteins. We know exactly what we're dealing with. It's not a complex mishmash of a cell. It's fast. We can do 10 repeats of a single condition in about 2 minutes. And it gives us kinetic information. How quickly do these molecular processes start and stop? And as like proof of concept of this working in our hands, we used a pore-forming antibiotic called gramsidin to see if we could see how it moves ions across these liposome membranes. We loaded up liposomes with the gramsidin antibiotic in a high potassium environment and then mixed them with the quencher with that high thallium environment to allow for potassium to efflux and thallium to influx. And what we can see here is that while control liposomes have stable fluorescence over time, the gramsidin-containing liposomes have rapid quenching of fluorescence on the order of 20 or 30 milliseconds or so. This is a fast molecular process. I think of it as like a bullet train of ion transport. There's not a lot of molecular rearrangement that needs to happen to allow this to go forward. We then wanted to turn this technique to studying APOL1 proteins. And so we expressed and purified APOL1 proteins. I'm happy to show size exclusion chromatography if people really want to see that, but I figured you might not. And embedded them into liposome membranes to study their ion transport activity. And we did that with the same pH step process we know is required to get APOL1 into its functional state. We associated them with membranes at low pH and then raised the pH up into the low 7s to look at their activity. We started with these pre-activated liposomes. Some of them are going to have open ion channels, some of them are going to have closed ion channels, but just wanted to see if we could see fluorescence changes here, indicative of ion transport. And we did. You can see here that APOL1 containing liposomes quench their fluorescence a lot more than control no APOL1 protein, just detergent containing liposomes. And adding a non-specific cation channel blocker like lanthanum or gadolinium blocked at least part of that ion transport. This was before we had the cool Vertex molecule available to us. I want to go back and do these with that. However, this is really slow. I was showing you time courses of like a quarter second on the bottom. Now we're looking at things that are 100 times slower, taking 3, 4, 5, 6 seconds to reach maximal fluorescence quenching. I think of these much more like the Green Line in Boston, which is incredibly slow. There's really not a Chicago analog because public transit is so functional here. So we then wanted to adapt that stop flow setup to look at how many of those molecular steps are happening for APOL1 to start working. This requires a more complicated diagram, but is not that much harder to understand, I promise. We start with those so not yet activated liposomes at that low pH. They have the APOL1 associated with their membranes. They have the ANTS fluorophore inside of them. They just haven't seen that higher pH condition that gets them to their active state. We then mix them with a base that brings the pH up and gets those ion channels to their active state, and they enter this delay loop. We can give them a little time to hang out there before we finally mix them with the quencher molecule that allows us to assess how many of those ion channels are open or closed. So what does that show us? In this, you can see that we had APOL1-G0, so low-risk variant containing APOL1 liposomes, and they show fluorescence quenching. They show more rapid fluorescence quenching than we showed before. These are on the order of 2 or 3 seconds. And that kind of makes sense. These are primed to open liposomes that we then open all together and look at all the ion transport all at once. There hasn't been time for them to desynchronize and work at different times. At the same time, they're still pretty slow. And how slow are they? We can actually use a stretched exponential function, fancy math, to fit this curve and get rates of transporter activation. At different amounts of delay time, at different amounts of time activating at that higher pH before entering the optical cell, the rates of ion transport are somewhere around 1 per second, and they're stable to going up slowly over time. This is really weird for an ion channel. Like a normal ion channel, like the potassium channel, has rates that are 80 times higher or faster than this right when it starts. And those rates decrease over time because the ion channels inactivate over time. Here we're seeing no evidence of inactivation over these timescales, but very slow activation processes in the first place. We also did this with the G2 variant containing liposomes and see more fluorescence quenching here than we did with the G0 variant containing liposomes. The numbers we have here are not as many as the numbers we had in the G0, so you're going to see some bigger error bars. But these rates are faster than the G0 variants at all those time conditions, but still very slow by absolute ion channel standards, still 40 times slower than normal potassium channels. I will say that these rates, if they really are double the rates of the G0, kind of makes sense with that previous data that shows the G1 and G2 variants work about twice as well as the G0 does. So based on these assays, I think we can say a couple of things about this transport activity. APOL1 allows cations to move across membranes, and this activation, it's activated by pH increases, but the activation is really slow. We're still seeing activation at 5 seconds. We don't show any inactivation on that kind of time scale. Again, really slow to inactivate transporter. And the high-risk variant proteins are activating more quickly than low-risk variant, but still very slowly. I think understanding how these transporters look, how they work, really requires us to get a more molecular level understanding of how many of these proteins are hanging out together at the same time to do this job. And so I'm going to turn to that next. Questions on the stop flow stuff, though? Yes, Sue. Yeah. Yeah. That's a good question. I think even though this is slow on a protein timescale, it's still like a reasonable length of time on a living thing timescale. So I think even slow activation in a lysosome and trypanosome, we could see this. And I think with endosome recycling in a podocyte or some other kind of cell also could be reasonable to see this kind of speed. Mm-hmm. Yeah. 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 So Sue's question is a really good one. That about 15 to 20% of people who have two high-risk APOL1 alleles are going to develop kidney disease over their lifetime. The rest of people probably won't. So what's driving those people to get kidney disease and not all 100%? APOL1's a super interferon regulated gene. And so the thought is really that some second hit, some inflammatory state, I don't know that there are one-hit diseases anymore, but some inflammatory state drives interferon production and APOL1 expression. And that the thought is that really makes enough of these ion transporters to do their job. Even if you overexpress the G0 variant to one or to twice as much as the G1 or G2 variant, it doesn't seem to be as cytotoxic. So yeah, so there's something else going on that's, I think, different between the G0 and the G1 and 2 activities. Yeah, yeah, and I think the idea is that when you put this small molecule into people, that it calms down whatever self-propagating process is going on. It kind of breaks a cycle. But I know that's really hand-wavy and not a good answer. So yeah, we need to know, and we don't know. Yeah, Vikram. Yeah, in kidney cells and podocytes. I'm not aware of that being a main site of injury. I'm going to turn to Jenny too and see if you have any thoughts on lysosomes in podocytes. I don't think that's where people are really fighting about as a major site of APOL1 action. I think the main fights are plasma membrane, mitochondria, ER as locations, more than lysosome. Yeah, yes, but it depends on whose model system you're working in. So I don't know that I can say across the board that there's one specific true answer. Yeah. So in trying to understand how many of these proteins there are working together in a complex, we used a technique called multi-angle light scattering to look at the size of these proteins in solution. To do this, you pass your sample through a chamber with a laser beam shooting through it. And if you have a big protein, it bounces more light off of it. If you have a small protein, it bounces less light backwards off of it. And based on the pattern of light scattering, you can calculate back the size of your protein. This works well for things like BSA, which you can see in its monomer and dimer forms, as well as hemoglobin, which is a good control for everything. And when we did this with our protein that we expressed and purified, we saw that it hangs out as a monomer. You can see the peak on the far left. It shows 42 kilodaltons of protein, exactly the APOL1 size that we know this protein is, and 70 kilodaltons of detergent hanging out, stabilizing it in solution. The other peak that's over here is a 33 kilodalton peak that's only detergent molecules, extra detergent. So these proteins that we make are monomers floating and tumbling in solution. Although this is a really great technique to look at molecular complexes, it requires these molecules to be floating and tumbling, and we can't use it in proteins that are embedded in lipid membranes, which is the place we think this protein is most active. To do that, we had to do turf microscopy, and actually a couple of us were at the microscope facility open house, and they actually have a really nice setup for this and a really nice description of how this works. This is a technique that takes advantage of a unique property of light, which is that when you reflect it off of surfaces, most of it comes back to you, but a small amount doesn't come back. And at the right angle, this laser light here, when it bounces off the glass fluid interface, right where that round circle is, most of the light will bounce back, but a small amount will illuminate the part of the fluid sample that's closest to the glass surface, about 100 nanometers of depth or so. And that really gives you exquisite XY resolution and drops out everything in the Z dimension. You don't see stuff that's farther back than that. How's that useful? Because if we have fluorescently labeled molecules, either with a protein or a chemical dye, and those molecules are up against that surface, we can see those spots of fluorescence and count how many fluorescent molecules there are per spot. Because fluorescence bleaching is a random process, you just crank the laser power and count the number of steps it takes to bleach, and then you know how many fluorescent molecules were in your spot to start with. So we're able to do this with APOL1. We made a single cysteine mutant. APOL1 doesn't have any cysteines, but we put one in and at the far end terminus away from the action and labeled it with a fluorescent chemical dye. We embedded it into the liposomes that we had made, just like for the stop flow assays, and then flowed those liposomes over glass cover slips. Liposomes kind of splat onto the surface of a glass cover slip when they see it, and they make this nice bilayer just along the slip. It's really perfect for this technique because you get most of your protein embedded in a lipid membrane right at that surface so you can look at it. And then we took it to a local collaborator who I met through my son's birthday party, who was able to look at these on a turf microscope. You can see here, so that's empty liposomes. There's kind of a little bit of blinking, but goes away. That's kind of what background signal looks like here. In contrast, when we have fluorescently labeled proteins, can you guys see the little dots there? Okay, so the dots are protein complexes with fluorescent dyes attached to them. So we're able to see those, and we can see them dim and blink off over time. We also see that with G2-containing liposomes. And I think here, when I look at it, I see a little bit more dimming and brightening, but you don't have to take my word for it. So we go through and look at each one of those fluorescent spots and watch the fluorescence change over time. In the empty liposomes, we see single-step photo-bleaching and of short duration of lifespan, really consistent with a background fluorescence of the lipids that we're looking at. That's really true of most of the spots that we can find in that picture. We see some of those background events in the G0 liposomes too, but when we look at those bright spots, we see other things. I think I'm not going crazy looking at these if I tell you that there are multiple steps in these traces. You can imagine two fluorophores blinking off in each of these traces, or blinking on and then blinking off in these traces, suggesting that these G0 proteins are forming oligomeric complexes together. In the G2-containing liposomes, we see the single and two-step photobleaching events, as well as some other pretty interesting looking traces with multiple steps to them. And I know I'm showing a selection of data, this is anecdata, but these traces are a little painful to do by hand. You have to find each dot and really pull the data from each one. We started working with a biophysicist I started working with a biophysicist at Northwestern, who is a better pipeline to analyze these data a bit more systematically. And I think that's going to help us get through more of these traces, get better answers at the oligomeric state of these proteins in membranes. So at some point between being in solution and being in its transport active state in lipid membranes, we see this APOL1 oligomerize. It goes from a monomer to some kind of oligomerization state, TBD. We don't know if that oligomerization event occurs right when it binds to the membrane or when the pH rises to form that active state. We don't know if it's reversible with pH drops. And we don't know if that could be a target for future APOL1 therapies. Could blocking this oligomerization event prevent the ion channel from ever forming in the first place? And could that be another way to get at this problem for people who might not respond to channel blockers? Questions here? I love stopping for questions. I think that's helpful. Yes, Sue? of G0 and G1, G2 variants. Oh, yeah. So it would be so one nice thing about these in vitro reconstituted systems, we can just mix G1 and G2 proteins together and see what we get. I think having an oligomeric state going in helps, because then you can get a sense of how many of each subunit would you expect in a complex to start with just statistics baseline. There are some really nice old school papers where they derive it from the kind of stop flow data. We haven't been able to do those yet. Jenny, is that a hand? Yeah. So the question is, I'm looking at APOL1 proteins alone by themselves in lipid membranes. Do we think APOL1 hangs out with friends in the cell to have this activity? Or do binding partners amp up transporter activity? And I think that's a really great question, and a question that's hard to answer in a cell-based system, like a real living thing. What's great about these in vitro approaches is you can add individual components back and figure out what effect that has on oligomerization or on ion transport. So that's not stuff we've done. I think that's on a future direction slide somewhere else. Yeah, it's a great question. Okay, so where are we going from here? I think there are a couple of low-hanging fruit things that we can do given that we have these techniques in hand now. It's easy to assess other ions conductance with this stop flow assay. They make lots of great chemical dyes that are quenchable by chloride or quenchable by calcium. And we should be able to see ion transport for each of those, see what differences there are for each ion. We can also look at effects of lipid composition. Each of those lipid membrane-bound compartments has different lipids in them. Mitochondria have cardiolipin, plasma membranes and ER are different from each other. We can see if there's one of those lipid membranes where these proteins are more active. We can also use small molecules like inaxaplin to block the protein activity and see if it affects other ion conductance besides that bulk thallium assay that's in the paper. We really don't know if it affects chloride at all or calcium at all or anything else. And finally, I think comparing the conductance properties of low and high risk variants is clearly the next step here too, but just don't have that to present at this point. An APOL1 structure would provide a lot of this information, and that's a major goal that I have. I think it would show us what the pore itself looks like, what the oligomerization state looks like physically, as opposed to these more indirect methods getting at it, and show us where the differences between the G0 and G1 and G2 variants are. This has been tricky for me and has been tricky for a lot of other people before me. But I think we're making progress in the right direction here. We're expressing these proteins now in hex cells and eukaryotic cells where they fold more normally. And we're working with a structural biologist at HMS who's helping give some more guidance on the best ways to go forward here. And then leaving the test tube, as much as I love working with one protein at one time in one tube, I know that isn't where this disease happens. I think having good purified proteins to work with lets us figure out what APOL1 binds to. We can use that protein as like a bait to go looking for binding partners and figure out what APOL1 hangs out with in health and in disease using real patient samples. If we're able to isolate these currents from each other and get small molecules that block a single current and not the others, we can also figure out which of these conductances drives disease progression, at least in animal models. And then oligomerization. I come back to this because I think it helps us answer the question of where does the APOL1 work? We can target dominant negative, like poison pill APOL1 constructs to a specific compartment and sort of site specifically knock out APOL1 activity in the mitochondria or at the plasma membrane and see, does it still have those cytotoxic effects? And that would really let us answer these long-standing fights about where the APOL1 is working and where it's driving cell injury from. So to wrap up, I've shown you some of the background about how we identify these high-risk APOL1 variants and that they contribute substantially to the high rate of advanced kidney disease in people of recent African ancestry. And although we don't have consensus around how these gene variants cause disease, I think most people are building around this idea that ion transport plays a role, especially driven by the ion transport blocking activity of these small molecules that we see. I've shown you that APOL1 proteins can form slowly activating oligomeric complexes that allow cations to move through them. And we know that this cation transport activity is a target for future therapies. And more broadly, I think studying proteins in reconstituted systems like this really lets us answer questions about mechanism that we can't answer any other way, and really lets us get better answers to questions about disease and about treatment. With that, I'm happy to thank folks who've supported me in doing this work, Martin Pollack, my mentor at Beth Israel, and two great research assistants I've had work with me over the last 2 years, as well as other collaborators and funding sources. I'm happy to take other questions. Yeah, Dr. Bhatia.
Fantastic talk, a very naive question. What can we learn from viruses like HIV?
Yeah, so what can we learn from how viruses activate APOL1-associated, or kind of trigger APOL1-associated kidney disease? The best way I understand that driving process is thought to be through interferon, through inflammatory states, although I know that's not a great answer. Most of the model systems we have use interferon as the inducer for APOL1 in mouse models. Interferon is not tetracycline. It has a lot of off-target effects. But that does seem to be a driving factor in HIV-associated nephropathy and these other acute viral illness-associated nephropathies thought to be part of the COVAN that folks were seeing in APOL1 high-risk genotype carriers as well. Yeah, Penelope.
Excellent.
Thank you.
Congratulations.
Mm-hmm.
So, and then I naive thought that I'm not do with the copy number. So I basically
Yeah, so interferon gamma definitely upregulates transcription and leads to more protein produced and leads to more cation transport in cell culture type models of APOL1 disease of APOL1 expression. What's still confusing to me is that even upregulation of the G0, the low risk variant protein, even if you upregulate it to twice as high as the high risk variant, you still don't get cell death and disease. And so there's something about some extra activity that the G1 and G2 variants, I think, have. I don't know if that's just ion transport, if there's some capped amount of ion transport that G0 can do even if you have lots more copies. I'm not sure, but I think copy number is definitely part of it, but I don't know if it's the whole story. Yeah, tomorrow. I'll come back to you in 1 sec, I promise. Yeah. Yeah, that's the method I've wanted to use and tried to use here. Getting beautiful looking samples to move forward with cryo-EM is tricky with a poorly behaved protein, which is the protein that I'm working with. But some of that comes from the fact that the protein is processed and hangs out in different compartments. Right now, I'm working to move the protein to the plasma membrane all at the same time and isolate it from there so that we can go forward with something like cryo-EM. But yeah, that would be that's the goal. Yeah, over here.
Thank you so much.
Oh,
Have
sure.
you seen any like calcium channels being variants?
Yeah, the calcium channel work I've seen, and Jenny is probably more up to date on this too, is looking at calcium sensitive dyes that folks can use in cell culture, and seeing that the G1 and G2 variant expressing cells have more calcium influx and more cytotoxicity at 24 or 48 or 72 hours after that. Do I think this protein directly regulates other calcium transporters? It's a good question. I don't know. I think understanding its binding partners would be really helpful to this, and then being able to build up that complexity is a really nice way to better answer that question. And I should say I'm comfortable with long, awkward silences. I taught Kaplan MCAT for a while, and the rule is if you ask the any questions question, you have to wait 7 whole seconds of silence until you get to move on. So again, but I've gotten lots of great questions. This is great. Yes, Sue. Sure. Yeah. So I missed the poster for that. It was an oral abstract or was it a poster? I forget if I missed it. Yeah. I think, so having not seen it, the thoughts I have about it would be, I think there are lots of ways to target this protein's activity. I think not letting it get expressed in the first place is a really good way to do that. At the same time, just scalability wise, we're still in a world where small molecule drugs rule the day. So until that kind of stuff becomes more widely available, I think we're stuck with figuring out how we can change protein activities with small molecules. Awesome. Well, thank you all so much. I really appreciate the engagement.